here we're going to kind of build on the complexity of understanding the two divisions of the nervous system. And so it's really important that you've got the anatomy straight, right? Because we're going to build on the types of fibers, the neurotransmitters associated, the types of receptors that are corresponding. So I really want to urge you, if you don't have the anatomy down, please go back and review it. Make sure you understand the length of the neurons, make sure you understand where they are located physically, right? Thoracolumbar versus sacral, versus craniosacral, right? Brainstem and sacral, and just have the anatomy down packed so that when we add this layer, it's even easier for you to make those connections. So as a part of the autonomic nervous system, we've got neurotransmitters, we've got receptors, right? We kind of talked about this loosely when we talked about the synapse, but here we're going to get into the specifics of the synapses and these players as it applies to the autonomic nervous system. So we've got cholinergic receptors, and that word cholinergic means that these receptors only bind acetylcholine. Acetylcholine is one of the most prominent, the most abundant neurotransmitter in the body. And so it only binds to receptors that are deemed as cholinergic receptors. This is also going to tie into the fast versus slow receptors, right? If you remember that from our last unit when we talked about the synapse as well. And so we'll clarify this later on, but you can go ahead and make a note in your own notes here that cholinergic receptors are always fast, okay? Always bind acetylcholine, always fast. The second type of receptor that we encounter in the autonomic system is adrenergic receptors. Okay, these are always slow or metabotropic receptors, and they only bind norepinephrine, epinephrine, or dopamine. Okay, so they cannot bind acetylcholine. But they do have some options as to what they can bind. They can bind norepinephrine as a hormone or a neurotransmitter, right? It's kind of the same chemical to the body. The route of transmission is what differs. So a neurotransmitter will be at a synaptic junction and a hormone will be traveling in the blood. But adrenergic receptors can bind either of those. It can also bind epinephrine, which is kind of the cousin of norepinephrine, and we'll talk about how they are slightly different a little bit later on. And then it can bind to a lesser degree dopamine, okay? And so these are the crucial differences between cholinergic receptors and adrenergic receptors is what they can bind, and then whether or not they're fast or slow, kind of connecting our understanding of fast and slow receptors from our previous discussions, okay? Let's think about the neurotransmitters here for a bit. So the neurotransmitters are going to be different depending on which neuron we're looking at. And this is why it's so important to have the anatomy down. So all of the preganglionic neurons, whether they're in the sympathetic division or the parasympathetic division, they always release acetylcholine, and they only release acetylcholine, okay? Huge, huge rule that is never, there's no exception to this rule. All the preganglionic neurons always release acetylcholine. On the parasympathetic side, only the postganglionic neurons release acetylcholine. So the sympathetic side, the postganglionic neurons are going to release a different hormone. And so only on the parasympathetic side do the postganglionic neurons also release acetylcholine. So you can kind of already make a connection here, right? That both parasympathetic neurons, preganglionic and postganglionic, always release acetylcholine. In the sympathetic system, the preganglionics will also release acetylcholine, but the postganglionics will release something else, okay? In the sympathetic system, the postganglionic neurons release norepinephrine, okay? And I've got two asterisks here because this is the sweat gland exception that I was alluding to earlier. So usually, postganglionic neurons in the sympathetic division release norepinephrine, but on sweat glands, the postganglionic neurons of the sympathetic system that innervate sweat glands release acetylcholine. Small exception to the rule for the sympathetic postganglionic neurons, okay? And then finally here, we can also think about hormones that are released from the adrenal medulla. Although they're pretty much the same molecule, because they're released in the blood, we're calling them a hormone, but they will also bind in the very same way that our norepinephrine does to adrenergic receptors. I'll talk about that, but we can kind of consider them as the source being different, right? One is a hormone coming from the adrenal, one is a neurotransmitter coming from the postganglionic neuron, but their action is going to be the exact same, Okay, so let me kind of just reiterate, clarify that here, and kind of connect the anatomy again. So parasympathetic system, long preganglionic neuron, that preganglionic neuron always releases acetylcholine. Short postganglionic neuron, that postganglionic neuron also always releases acetylcholine. Sympathetic system, short preganglionic neuron, that neuron releases norepinephrine, okay? We also want to think about the other options for synapsing of that preganglionic neuron. If it synapses onto the chromaffin cell, it's going to still be norepinephrine or epinephrine, but here it's going to be a hormone because it's going to enter the blood. If it's at a synaptic junction, it'll just be norepinephrine. As far as the postganglionic wait, hang on here. I just made a big mistake here. So sympathetic nervous system, let's start that again. Sympathetic nervous system, short preganglionic neuron releasing acetylcholine, right? We said all preganglionic neurons always release acetylcholine, okay? So please excuse my error earlier. So sympathetic nervous system, preganglionic neuron releases acetylcholine, and then the postganglionic neuron is where we've got those options now. We talked about it being an adrenomedullary chromaffin cell, or it's synapsing onto that collateral ganglia. And so the release here is going to be norepinephrine as a neurotransmitter, or norepinephrine and to some degree epinephrine as the hormone. Okay, I spent quite a bit of time here because this is something that's usually very confusing for students, and it really usually relies on you having an understanding of the anatomy here as well. Okay, all right. I encourage you to draw this out the way it's drawn out here. It's really going to help you make connections here. So this is showing only the sympathetic nervous system, the sympathetic division. And because we had 3 routes of communication for this branch, we can think about 3 options for the neurons here. So we've got a preganglionic neuron, which is short in length, synapsing onto the sympathetic chain here. This neuron will always release acetylcholine. It's going to bind to a nicotinic cholinergic receptor. We talked about the fact that acetylcholine only binds to cholinergic receptors, right? And then this is going to have a second neuron, which is the postganglionic neuron. Because this is in the sympathetic system, the second neuron will not release acetylcholine unless it's going to a sweat gland, okay? So it's usually going to release norepinephrine, which will bind to an adrenergic receptor on the effector organ itself. The second option, or the second route for communication here, was we've got a preganglionic neuron. It synapses onto the adrenomedullary chromaffin cell. And then from the adrenomedullary chromaffin cell, we've got the release of epinephrine. Here it's going to be a hormone released into the blood, and then that will be able to bind to adrenergic receptors on any effector organ. And that's where we get this diffusion of the response. Because this hormone is going to be released into the blood, it's going to encounter any adrenergic receptor in its path, and it's going to bind to that adrenergic receptor, and that's going to bring about our sympathetic response on all of the body's tissues that are encountered by this hormone. And then the third route of communication was to a collateral ganglia. So we've got, again, a preganglionic neuron releasing acetylcholine, that binding to a nicotinic cholinergic receptor. And I'll talk about the difference between nicotinic and muscarinic a little bit later on, 2 types of cholinergic receptors. But right now we're going to keep it general, and we're going to consider that acetylcholine is binding to a cholinergic receptor, and that cholinergic receptor is on the postganglionic neuron, and then that postganglionic neuron will release norepinephrine, okay? Really important image, guys. I encourage you to take this image, try to recreate it on your own from your understandings, so not looking at it or just memorizing it, but try to recreate this image from your own understanding. And I encourage you to also combine this image with that first schematic that I talked about. So be able to put those 2 concepts together. So the anatomy, and then add on the neurotransmitters and the receptors. So I'm getting a question, what happens if there's no cholinergic receptor? So if there's no cholinergic receptor, then there's a problem. There's a big problem for this for this response. There needs to be a receptor in order for the signal to carry through. And we'll talk about, so for example, myasthenia gravis is an autoimmune disease where we have autoimmune destruction of some of these nicotinic receptors, more involving skeletal muscle. And so it brings about paralysis, right? The signal cannot be transmitted. And so there are going to be huge consequences if there's disruption of the receptors, or if for any reason there's not a receptor present to accept or transmit the signal. Okay. And kind of going way back to the synapse and talking about the conduction, action potentials, the transmission of electrical energy to chemical energy. And so there needs to be a receptor for that chemical energy to be picked up as a signal on the adjacent neuron. So second question here, the first and third examples are the same besides the length of the neuron. Yes, that's correct. The length of the neuron, as well as, more importantly, the location of the ganglia. Okay, so the first example was a ganglia that's a part of the sympathetic chain. The third example here was a ganglia that is a collateral ganglia, so it's not a part of that sympathetic chain. Okay. Really good questions coming up here. If you've got more questions, you can keep them coming in the chat. Okay, so let's look at the parasympathetic system and kind of contrast that anatomy and the neurotransmitters here. So as we can see, things are much more straightforward in the parasympathetic division. We've got one kind of schematic, one model for how things are transmitted. The preganglionic neuron, longer neuron, synapsing onto a ganglia that's close to the effector organ or effector cell. And these preganglionic neurons are always going to release acetylcholine, so it's not much of an exclusion here, because even in the sympathetic system, preganglionic neurons always release acetylcholine. But on the postganglionic side, the parasympathetic neurons also release acetylcholine. So one way to kind of just simplify this for you all is that all parasympathetic fibers, whether it's preganglionic, postganglionic, they always release acetylcholine. Okay, the type of receptor they bind to is where there's a distinction. So although they're both cholinergic receptors, one is a nicotinic receptor, one is a muscarinic receptor. And again, I'll get into the distinction, the differences between those two types of cholinergic receptors a bit further down. Okay. Oh, so here we go. So distinguishing between nicotinic cholinergic receptors and muscarinic cholinergic receptors. So we said earlier that nicotinic cholinergic receptors are always fast receptors. They're ionotropic receptors. The channels are the receptors. We talked about this. Oops, excuse me here. Okay. We talked about this whole fast response, right? So nicotinic cholinergic are always fast receptors. Muscarinic receptors and all adrenergic receptors are always metabolotropic receptors, so slow receptors, okay? This is where they're coupled to a G protein. We've got the whole second messenger pathway going on, some type of response in the cell that could open up another channel or could bring about cellular response via some secondary response via the second messenger, okay? So again, a very important distinction. I get students mixing this up quite a bit. All nicotinic cholinergic are fast. All muscarinic cholinergic are slow. All adrenergic, which are the ones that bind norepinephrine, epinephrine, and dopamine, those are always metabotropic or slow as well. Okay. So let's talk about the location of the receptors. And again, I'm going to be jumping back to this image where it kind of all is illustrated here and makes sense. Let's talk about what receptors are found on what neurons. So nicotinic, on the postganglionic neurons, because all the preganglionic neurons, whether it's sympathetic or parasympathetic, because all of those neurons always release acetylcholine, we always have a cholinergic receptor on the postganglionic neuron, on the soma of that neuron. And let me jump back here to point that out. So because this neuron releases acetylcholine, this soma has to have a cholinergic receptor to bind that acetylcholine. It only makes sense, right? Same thing here. On the chromaffin cells, we've got nicotinic cholinergic receptors. On these postganglionic cells, here's postganglionic, excuse me, talking too fast. On these postganglionic neurons here, we also have receptors as well, okay, the nicotinic cholinergic receptors. So because these neurons always release acetylcholine, we always have cholinergic receptors. Let's go back here. The effector organs now are going to have different receptors depending on what division we're involved in. So if it's an effector organ in the parasympathetic division, it's going to have a muscarinic cholinergic, not a nicotinic. And then if it's in the skeletal muscle system, it's going to always be a nicotinic. As far as the sweat glands, we talked about sweat glands being that one exception to the rule on the postganglionic neurons in the sympathetic division of the nervous system. Although this is in the sympathetic division, these neurons still release acetylcholine, and so they're going to bind to a muscarinic cholinergic receptor. All righty. Okay. Let's talk about the adrenergic receptors here for a bit. So we've got two main types of adrenergic receptors. And just to kind of remind ourselves here, adrenergic receptors are the ones that only bind norepinephrine, epinephrine, or dopamine. They have differing affinities depending on exactly which type. So some of these prefer epinephrine, some of them prefer norepinephrine. And so we're going to talk about the main subclasses, but then we're going to talk about the different subtypes and their differing affinities for those different neurotransmitters or hormones. Hang on here. Okay. So the two main types of adrenergic receptors are alpha receptors and beta receptors. These are all metabotropic, so all coupled to a G protein, all producing a slow response. And then we've got different subtypes underneath each of these. So alpha receptors have alpha 1, alpha 2, beta receptors have beta 1, beta 2, and beta 3. So let's look at the alpha adrenergic receptors first. They're mostly in, they're the most common receptor that we see in the sympathetic division. They're located on most of our organs, the effector organs we talked about. So blood vessels, usually, they're usually excitatory. So that we're going to talk about two different types of effects that can be brought about on the effector organ. So we can have an excitatory effect. We can also have an inhibitory effect, which we'll get into a little bit later down after we look at all the receptors. But the alpha adrenergics are usually an excitatory. So think about constriction of smooth muscle as an excitatory effect on the tissue. And then the affinity here is that they prefer norepinephrine. So all of the alpha receptors, alpha 1, alpha 2, they have a greater affinity. They prefer norepinephrine over epinephrine. Okay. And then looking at the beta receptors here, again, more generally, we'll get into the specifics in a bit, but all of the beta receptors activate cyclic AMP as their second messenger. We'll see that the alpha receptors have two different options for second messenger, and then the affinities here are going to vary depending on the subtypes. So if we look at beta 1 receptors, the location of beta 1 receptors are usually on cardiac muscle and kidneys. The effect on tissue is usually excitatory. And then the affinity here is equal. So beta 1 receptors don't have a preference as far as norepinephrine versus epinephrine, and they have an equal affinity for both of these neurotransmitters or hormones. And I'm going to keep saying neurotransmitter or hormone so that you can think about the adrenal medulla option, right? So it can be norepinephrine as a neurotransmitter at the synaptic junction, but also as a hormone in the blood. As far as beta 2 receptors, usually on blood vessels, beta 2 is also in the lungs. Although it's not listed here, you can kind of put that in your notes. So beta 2 is usually on lungs, as well as some of the smooth muscle in our blood vessels, usually an inhibitory effect on the tissue. And so for example, in the lungs, we see that this causes bronchodilation, and that's considered an inhibitory response, whereas constriction is going to be an excitatory effect on the tissue. So again, beta 2, usually inhibitory, and then the affinity here is greater for epinephrine than norepinephrine. So kind of a flip-flopping of what we saw with the alpha receptors in general. So beta 1, equal affinity, beta 2, greater affinity for epinephrine, meaning that beta 2 receptors have a higher binding to epinephrine than norepinephrine. And then beta 3, usually on adipose tissue, usually also excitatory, and then also having an equal affinity, just like what we saw with beta 1 receptors.